Hello, everyone. If you've tuned in for the Underwater United States, brought to you by the Woods Hole Oceanographic Institution and the Mid-Atlantic Regional Council on the Ocean, you're in the right place. We'll get started in just a few minutes. Once again, if you're just joining us, you're tuned into the Underwater United States, brought to you by the Woods Hole Oceanographic Institution and the Mid-Atlantic Regional Council on the Ocean. We'll get started in just a few minutes. People are still joining us. Once again, if you're just joining us, you're tuned into the Underwater United States, brought to you by the Woods Hole Oceanographic Institution and the Mid-Atlantic Regional Council on the Ocean. We'll get started shortly. Hello and welcome to the Underwater United States, exploring the life and ecosystems in the U.S. exclusive economic zone. My name is Ken Costa. I'm the Director of Research Communications with the Woods Hole Oceanographic Institution, and I'll be your host today. We're going to jump right in. First, I'd like to introduce Kisha Santiago-Martinez. Kisha is the Deputy Secretary of State with the New York State Office of Planning, Development, and Community Infrastructure. And in her position, she's at the forefront of efforts in New York State to advance sustainable, equitable, and resilient development, and to expand New Yorkers' access to offshore wind energy, to improve climate resilience and mitigation at regional and local levels, and to protect and restore the state's coastal and ocean ecosystems. As the head of Office of Planning, Development, and Community Infrastructure, she oversees a wide range of policy initiatives and programs, including the New York State Coastal Management Program, and she serves on the Governor's Climate Leadership Action Council Executive Steering Committee. And most importantly for us today, she's also the current chair of the 5-state Mid-Atlantic Regional Council on the Ocean, which she's going to tell us a little bit more about. Kisha?
Great, thank you, and good afternoon, everyone. Through our wonderful partnership with the Woods Hole Oceanographic Institution, WHOI, Marco is so thrilled to present this webinar, Underwater United States, Exploring Life and Ecosystems in the U.S. Exclusive Economic Zone, EEZ, Corals and Canyons. In particular, I'd really like to thank Dr. Tim Shank, Associate Scientist and Deep Sea Biologist at WHOI, who will be presenting this absolutely stunning material. I'd also like to thank the Gordon and Betty Moore Foundation, whose continued support allows Marco to collaborate on projects like this, which yield important data that can be used to inform and guide ocean policy and decision-making. I have the great honor of serving as chair of Marco, which is a regional ocean partnership for the Mid-Atlantic. Established in 2009 by governors from New York, New Jersey, Delaware, Maryland, and Virginia, Marco is an interstate collaboration that addresses shared regional priorities and provides collective voice on important ocean issues that transcend political and geographic borders. Dr. Shank's research on mid-Atlantic corals and canyons, which you will learn about during today's webinar, has been an important contribution to building an understanding of deep sea habitats off the mid-Atlantic coast. His research is directly relevant to Marco's goals of helping states and other decision makers in the region maintain sustainable ocean resource use and healthy ocean ecosystems. Millions of people live along the mid-Atlantic coast and spend time recreating on its beaches and shores. Yet few people know that far offshore and deep underwater lie some of the most majestic and remarkable features in our ocean. Deep sea canyons run along the underwater coast of the mid-Atlantic and provide vital habitat to ancient corals and other unique species. Studying these fragile habitats and the hidden and unusual species that live in them is so important to understanding how decisions and daily behavior can impact little known but critical areas of the ocean far away and unseen. Poor water quality can be harmful to slow-growing aquatic life like coral. Climate change can disrupt water movement, and marine debris has even been found in these remote deep sea canyons. Over the next hour, we will cover some of these exciting findings from the 2019 Mid-Atlantic Deep Sea Canyon Expedition. You will learn about the important discoveries gleaned from these unique and poorly understood habitats, which will help raise awareness about their importance. Based on this exploration, our team has also created an ocean story and a story map, which can be found on Marco's website. Detailed information is also available on Marco's Mid-Atlantic Ocean Data Portal, and a link will be sent out to all members who are participating in an email after the webinar, along with some additional resources. Once again, I want to thank you all so very much for joining us today. This is really going to be an exciting virtual tour for some of Mid-Atlantic's most stunning deep sea canyons and coral colonies. With that, I'd like to turn it back over to Ken.
Thanks, Kisha. And I want to reiterate some of that story map that Marco has produced is really stunning. And I can't tell you how pleased we are to be working with Marco to organize this event today. And thanks also for Marco's support of Tim's work. It's really important to be able to shed light on the canyons and the marine habitats of the mid-Atlantic. Before we get started, I'd like to take just a quick minute to get a sense from where people are tuning in from today. If you joined us on Zoom, you should be able to see a pop-up poll appear on your screen shortly. There it is. asking you to indicate the state where you live. Most of these are the states that Marco encompasses, so I imagine we'll have quite a few from those, as well as from Massachusetts, where WHOI is located. If you're not from one of those places, you have just two other options. Click the one that's closest to you. While we're finishing up with the poll, I'd like to give you just a few tips on how to optimize your Zoom event experience today and how to participate. Later on in the program, our presenter will be taking questions from all of you. If you'd like to participate, please use the Q&A button in the Zoom screen and type your question into the window that appears. Most of you are probably more familiar with the chat feature of Zoom meetings, but we've disabled that today for the webinar. So please submit your questions via the Q&A button. If you're watching on the YouTube live stream on the WHOI or the Marco channels, you can submit questions directly into the comment box and someone from our team will relay them to me. We very often get hundreds of questions, but we'll make every attempt to get as many of you as possible. We've had over 500 people register, so we may have a pretty active Q&A session. I also want to let you know that we'll be recording the event, and the recording will be available later on the WHOI and the Marco YouTube channels. And let's see, the results are in. And Kisha, you'd be happy to know that New York is showing a distinct lead in in people tuning in. Okay, let's get started with the main event now. Tim Shank is a deep sea biologist and an associate scientist in the biology department, and also the former director of the Ocean Exploration Institute here at the Woods Hole Oceanographic Institution. He's known for his research on the ecology and evolution of animals that live near deep sea hydrothermal vents, on seamounts, and in underwater canyons and trenches. He's conducted more than 60 scientific expeditions in the Arctic, in the Atlantic, in the Pacific, and the Indian Oceans, and he's completed more than 50 dives in the research submersible Alvin. He's also conducted more than 100 dives with autonomous underwater and remotely operated vehicles, including making the first use of a one-of-a-kind remotely operated vehicle in some of the ocean's deepest trenches, including one expedition that I was fortunate to be on with him. He's also the author of the award-winning best-selling book, Discovering the Deep. So he is well-placed to tell us all about the canyons of the Northeast. So Tim, thanks for being with us. Take it away.
Thank you very much, Ken. I really appreciate it. And thank you to Kisha for all those great comments. It's true, I've had 28 years of exploring and conducting research in the deep sea. And I've been all over the world. I've been very fortunate. But I have realized that we have the deep sea right off our coast here. And it's been an amazing sense of energy and renewal of trying to understand the deep sea and all its glory and what it means to us and how it impacts us, that I'm so thrilled to be here today and add my welcome. Thank you for joining us here today. And let me share some things with you, some things I have never presented before. You're going to learn some new things today. And I'm really grateful to be here. It's also not just a single effort. There were people who conducted expeditions with me in the canyons, Dr. Martha Nizinski, Taylor Heil, and Brian Kidlin, and Matt Pody, and a lot of others. And the research vessel Bigelow was also instrumental in this. And there's engineers and technologists. Anyway, it's not a one-person show. And so I'll acknowledge them at the end, but I want to do that at the front as well. So anyway, I would love to get started. I always want to get started by telling people about what it looks like. I've tried to show them what it's like to be on the seafloor and how far these canyons are away from us. They're less than 100 miles in some cases. And so when the first mapping data came out only 8 years ago off our own coast, I made this animation to try to show that. And if we could run that animation, you can see here we've got Long Island and Cape Cod in the top left. And what we're going to do is I'm going to take you underwater and take you down to the canyons. Here's Long Island. We're going to slip underneath Cape Cod because it's so local to us here and go over Stellwagen Bank and the continental shelf, which are over now, relatively flat. And then we're going to dive over into the deep sea, into the canyons. When the glaciers were retreating 20,000 years ago, water, rivers carrying lots of sediment carved out these canyons. Sea level was much lower then. And you can see them all here. There's 90 canyons, over 90, between Canada and the Carolinas. They're astounding. They're massive. They go from a quarter mile deep down to 2.5, 3 miles deep. Here you're seeing some seamounts that are off our coast called the New England seamounts there too. But you can see them all the way down the coast. Just simply amazing. And they're right in our backyard. I think so few people realize, as I did not when I first started working on this, is that the amount of land underwater the U.S. has is 1.5 times that of the continental U.S. on land. It's simply amazing. And any of these canyons that I'm going to show you today could easily be a national park if it were on land. In many cases, the majority, I'd say, is that the Grand Canyon could fit inside these canyons, one of these canyons. So they're massive. And yet we know so little about them. We've only just begun to explore them in the last few years. So what I want to do now is get a close-up of some of these canyons, showing you the topography from the mapping that's been done. And so here you see this is bathymetry of the seafloor, with the red part being about a quarter mile deep and the bottom of the green part being about 1 mile deep. And so you see immediately there's these canyon structures that come out, and they have lots of topography, rough walls, buttresses, lots of hard bottom substrate there, because the currents in this area come from right to left in this image. And they sweep into the canyons, and they hit these canyon walls that are completely, I'll say, gnarly in their formation. And the currents whip around in these canyons and bring nutrients up from the deep sea up toward the surface. And similarly, the red part you see here, which is relatively shallow, like the heads of the canyons, this is where we have a lot of plankton. We have a lot of zooplankton, a lot of animal life and plant life that's using photosynthesis to generate a lot of productivity and bring carbon into the system. These animals die. And they fall to the seafloor as carbon. And so there's a lot of productivity in these canyons. And it's not just coming up and coming from the bottom and coming down from the top through currents, but it's also being fed by animal systems that are living there. It's amazing to me to learn that these are hotspots of biological diversity, these canyons, when it comes to whales and dolphins. They flourish in these hotspots of productivity. This graphic you see here, the New England Aquarium, shows that the head of the canyons, the top of the canyons, the red part you see, is loaded with marine mammals. Just in one day of surveying by plane, they see Rizzo's dolphins, 50 humpback whales, 120 fin whales. There's more than 13 species of whales they see, and more than 10 species of dolphins, all flourishing because of this nutrient-rich waters that are funneled into the canyons. So these canyons have a major impact on biodiversity and the life that lives there. It feeds everything, including humans. When we first came to North America, we really benefited from the fisheries, and I think we still do, from the productivity in these canyons. So to go to the next slide, in 2012, we initiated studying these canyons. They weren't even mapped yet. Through work with Martha Nizinski and others, you can see here the canyons we've identified in yellow along the margin of the East Coast here. And we studied for 2 years the northern canyons in the Northeast. And in 2014, we set our sights on surveying the Mid-Atlantic canyons, see what was in them. They had virtually been unexplored. And we didn't have a submarine or a really remote vehicle to go in there and do the first order survey. We needed a really reconnaissance vehicle. And so I want to click in now and show you just a little bit closer the survey areas that we use with this vehicle. Here's a zoom in to show you a couple things. Here are the canyons we looked at in 2014. There were 8 of them, 7 here in the Mid-Atlantic. And you see how close they are to New Jersey and to Delaware and to Maryland. I mean, these things are less than an hour and a half car drive from the beach, okay? So they're really in our backyard. They are our underwater America. They're ours as we have national parks or any lands anywhere. They're part of our country. And yet they remained unexplored until just a few years ago, or even mapped for that matter. And so you can see them here distributed as Washington Canyon, Accomack, Leonard. I'm just reading the names off because you're going to hear them again and again during this talk. Wilmington, Spencer, Lindenkohl, Colorado, and then many more minor canyons, smaller canyons. If we go to the next slide, I'll show you a subset of these that we looked at. So here you see, again, topography of the seafloor, and you see the continental shelf in red, basically flat, right? And then you go down into these canyons, Washington Canyon on the far left and Wilmington on the far right. You can see how these cut into the continental shelf. We call them incised canyons. They incise the margin. as opposed to like Accomack and Leonard you see in the middle there that just barely snig the continental shelf. And so when we first saw these maps, we said, I wonder if the productivity, the energy in these canyons is different. If the ones that insize into the margin, if they're more productive, because there's more of a channeling there. Channeling, you can bring down productivity from the shelf and more area for animals to live from the nutrients that are being brought up from the deep sea. And you can see how far reaching these canyons are. Even to the bottom of this image, you see the canyons continue on sometimes over 400 miles. And so they have a massive impact in transferring of energy and productivity in the deep sea. So that was our standing hypothesis. And maybe the incised canyons actually have more productivity, which may mean more life than these smaller, minor canyons. And many of these canyons are unexplored and unnamed, as you can see. If you go to the next slide, we'll show you the survey vehicle that we used. It's called TowCam. It's basically sort of a dope on a rope, but it's a little bit more intelligent than that. It's tethered to the ship, and we tow it behind the ship at about 1/4 knot, which is a modest walking pace for any human. It has a 24 megapixel camera looking down, batteries on the vehicle. It can measure temperature, depth, and it has flash strobe lights. It takes a picture every 10 seconds. And so we tow this along the seafloor for maybe eight hours at a time in these canyons, starting from deep and going up to shallow. It logs the images on board the vehicle. We bring it on board, we download the images, and then we analyze the images for whatever life we see in them. And so here's what I want to show you that we saw. The next slide, please. In these canyons, it was just amazing. We found really high diverse deep sea corals and sponge ecosystems in these canyons. And here's just one example. You can see the canyon sort of rock itself. It's like there's a wall here with a large buttress jutting out. And you see lots of these corals, these yellow fan corals, all different kinds. To date, we think we've discovered over 70 species of corals that live in these canyons. The next slide will show you just another example. You see they have this morphology. They're almost like trees jutting out from the walls. These are called bubblegum corals, which occur in almost like dense forests. They're really amazing. It's hard to get a sense in a still image of just the three-dimensionality of these corals and how they dominate. I think we have a short video here I want to show to give you more of a sense of what this place is like. Here you see another buttress like this, a slope on the margin of a wall here, just vertical walls that extend for hundreds of feet. If we roll that video, in the first image here, you'll see that we've even got over 15 corals of different species. It's just amazing. Yeah, and they seem to be layered. They're here because they want that nutrient-rich water that's flowing by. It's sweeping all the sediments off these rocks. These are mushroom corals, and they're not like shallow water corals, as I'll tell you in a minute. They don't need sunlight. They live in absolute darkness. Here are just scads of single groups of stony corals, lots of red crabs, all this vertical structure here for these corals to be on. You see this one jutting out. This is a bubblegum coral that's white. Just amazing, but lots of animals living on these corals is one of the things I'm most passionate about, is the life that these corals host. And here's a large hakefish that makes its home around these corals. They actually dig out, some of these species of fish dig out little holes that they live in and create more three-dimensional habitat for these corals and other animals. There's sea stars here, but you can see they're massive. They're maybe 6 feet more in some cases. And so they're just astounding. And we find these in virtually all these canyons, as I'll talk about in just a minute. And here's a close-up. You can see there's shrimp living on this paragorgia, a bubblegum coral, red one. You can just see the tentacles there. And this is what I'm going to talk about next, is deep water corals. But it's a really good illustration of the polyps that are on these corals. They're not shallow water type corals. They don't have zooxanthellae or symbiotic algae. They have feeding polyps that have tentacles on them that grab particles in the water. If you go to the next slide, I'll give you a description of these deep water corals that seem to dominate these canyons. These corals live in darkness. They're deeper than 50 meters or 150 feet. They love cold temperatures. If things get really warm, I'm thinking of 14 degrees Celsius. I don't know if that's 50 degrees Fahrenheit, something like that. They don't like it very much. They love to be in strong currents because that's how they feed. They need those nutrients that are coming around this organic material in the water column. It's coming by, so they don't have zooxanthellae like shallow water corals. They're super slow growing. something that we've dated a few of these corals in our scientific community, and they can be hundreds of thousands of years old, believe it or not. And one of the things I really studied here is that how they provide habitat for more than 3,500 species around the world. So they really are foundation species. They're ecosystem engineers in these canyon systems. There's four major types of corals in the deep sea. There are hard corals, like you've once seen you pictured here, that's white. And you see one here that's octocorals in the bottom left. They're soft corals. We also have black corals of different types. Well, I'll talk about these in just a second. And also sea pens. These are octocorals that actually love sediment almost exclusively and have a really hard, rigid axis and really fleshy polyps. If we go to the next slide, I'll tell you a little more detail about these corals. Here's an example of a stony coral. These actually build reefs, these colonial corals in the deep sea. And what they're stony because they have their skeleton on the outside and the fleshy parts on the inside, kind of, Anyway, the next slide will show you a soft coral where it's just reversed. That the skeleton's on the inside. This is a very small skeleton here, but there's a thin veneer of tissue over the skeleton, and then you have these polyps. And they're called octocorals because they have 8 tentacles that surround a mouth, and that is what the polyp is. And so they wave these tentacles out, they capture particles, bring it into the mouth. It's simply amazing. If you go to the next slide, I'll show you a black coral. And they have their polyps too, but they have 6 tentacles that make a polyp, but they're linear along a branch. They're not in a circle. That's pretty amazing. And so Tim, you're probably saying to me, Tim, this is not black, this is orange. Well, black corals can be yellow, white, red, pink. It's the skeleton that's black. And it's that unique skeleton they have that actually people cherish them for jewelry. In this case, a lot of the ones in the canyons are this orange color. And they also feed in the water column. If we go to the next slide, I'll comment something about the ecology of these corals. I mentioned that they're really slow growing. This is an example of one off Hawaii. It's in about 500 meters of water, or 1,500 feet. It's about 4 feet tall or so. And this was sampled, and it was dated, and it's the oldest living thing we've ever known in the ocean. It's over 4,000 years old. It means these corals are growing microns a year. Which is pretty amazing. And so it means that they're so slow growing that if you happen to disturb one of them, if you knock one of these over or cut off a branch or something, it's going to take a long time, centuries, for these things to grow back. And so we're always cognizant of that as scientists. We go out and we only take a small snippet so we can understand who it is and more about its ecology and its evolution. If we go to the next slide, this is something that's near and dear to my heart, is that these coral species serve as habitat. And this is where all the diversity comes from. And in coral ecosystems in the deep sea. And I've got this example here. I'm going to show you a couple here because I love them so much. Here are 3 different corals in the deep sea. There's one on the far left, one in the center that's yellow, and one on the far right that's kind of feathery. And each of them have a brittle star on them. And there's one down below too, actually. There's a white coral down below. It's a hard coral. And they each have a different species of brittle star living on them, yet they're only less than a foot apart, this host coral. And so somehow these animals have learned how to recognize a certain species of coral, settle on it, and spend their life there. These are very specific co-evolved relationships that are simply fascinating. We go to the next slide. Here's another example. Here's a yellow coral, a soft coral, that's got a brittle star on it that will spend its entire life there, we think. And somehow it finds it wraps its arms around that coral, it feeds in the water column, we think. Maybe it may clean off the coral and feed off material off the coral. And you also see an anemone here that also lives exclusively on deep water corals. It's pretty amazing. Next slide, please. We'll show you a crab that also is highly evolved to be and live only on this particular soft coral. It's got legs that have hinges on them that are just shaped right to fit the branch size of this particular coral. And it will live in that coral, and it will pick organic material off that coral and feed and spend its life there. We don't know how they find these places, how they've evolved to be here or anything. We're just noticing that they have these patterns of co-occurrence. Another example is in the next slide that I love. This is a black coral. It's a bottle brush shaped coral, and it has this crab on it. And every time we see this coral, it seems to have this little crab on it. The crab's about the size of a quarter. It's really small. And when we look closely, it usually has a male crab and a female crab on it. And every once in a while, it has a little juvenile crab on it, like it's a family unit. It's simply amazing. These corals may be fostering families in that way. It's one of the hypotheses that we have. But again, to show you that these are fantastically diverse and yet unique ecosystems on these corals. It's true too, in the next slide, I'll show you that fish also have a close relationship with deep sea corals as habitats. A lot of fish lay their eggs on specific corals. A lot of fish use them as nurseries, as habitats for juveniles. They hide behind them for refuge. There's many ways these fish use corals, and some fish are known to be around certain corals. And so they're integral to the canyon ecosystem and how it fosters life histories for different species. So everything's so tied together, integral in these coral ecosystems, and they're in these canyons. We go to the next one. It's going to show you my last example. And this is of one of my favorites. A few years ago, we discovered an egg on one of these corals. And we kept noticing more and more eggs only on certain corals. And it turns out it's a Dumbo octopus, an octopus that has these fins that flap as it swims. And before we discovered this, we had no idea of this part of their life cycle, of how they actually where the eggs are and how they actually hatch and what the hatchling comes out to be like. And we show a video here. I'm going to show you the one and only video of a Dumbo octopus hatching out of an egg that was laid on a coral singly by the mother octopus. Here it is, just freshly out of the egg in a bucket on the ship. And just a few minutes later, it starts flapping. I put it into a bowl, and it flapped around in this bowl for 10 minutes or so. And we conducted MRIs on this thing and learned that when it hatches out, it's got 3 to 6 months of yolk. It can live off that, or it comes out fully formed. It's ready to feed and be a full-fledged octopus. And so it was an amazing discovery that this happens. We've seen in the Pacific too now. We've seen these eggs around on corals now. We know what to look for. And so these intimate relationships, we're learning more and more about them every time we go out. And so the question became, are we going to see these kinds of corals in the mid-Atlantic canyons? What are we going to find when we go there? And I'm going to show you, if we go to the next slide, I'll show you some of the examples that we saw here. Here's Wilmington Canyon. It's one of those that incisors the margin, and it's a large canyon. You can see in the top left scads of these solitary hard corals along vertical walls, with lots of sponges. You can see a bubblegum coral on the top right extending out into the water column with other corals, soft corals around it. Octopus in the bottom left. Another really large soft coral in the bottom right. If you look close enough in the center, you'll see a white structure there. That's actually a plastic bag. And I'll talk about that in a little while later. But here, lots of diversity, lots of coral ecosystems here in Wilmington Canyon. If we go to the next one, which is Lindenkohl Canyon, another large canyon. On the right, you see soft corals living on the margins of the walls there too, flourishing, doing really well. If you look at the left-hand side, we saw a lot of this in Lindenkohl Canyon and Spencer Canyon, but these are skates that occur in large abundances on the margins of these walls, and we saw this a lot with synaphid-branchied eelfish, with hakes and other fish that they would aggregate along these walls. Truly amazing. We see next example in Spencer Canyon, another Mid-Atlantic canyon, close to Lindenkohl and Wilmington. On the far left, you're looking down now. At the top here, you see a line of soft, hard corals along the margins of that wall. My screen went a little bit blurry here, but anyway, there's lots of soft corals here on the right. I mean, all the canyons that we saw flourished with corals. So that's great. So that's good. That's the first question, all the corals in these canyons. The second question is, because these canyons have such a big impact on productivity and diversity, how they host the biodiversity that I was just telling you about, to help those other animals survive and even evolve, we really want to know the distribution of corals in these canyons. It's really critical to know. And so I'm going to walk you through this carefully here. If you go to the next slide, I'm going to show you this graphic, and please bear with me because it's the first time I'm presenting this, and I really appreciate your patience. But on the far left, you've got depth as it descends, 500, 800 meters down to 2,000. Remembering that 800 meters is about a half a mile, and 1,600 meters is about a mile. And then what I've got on the far right here, the different types of coral we looked at. We looked at 13 different types of corals in these images. There were over 46,000 images that we looked at in these 7 canyons. And so you don't have to worry about the coral types. Just look at the colors, the different colors. And what I've got here is a column underneath the word Wilmington. This is Wilmington Canyon, and it shows you the distribution, those colors do, by depth of the different types of corals. And I hope that makes sense. The colors represent different types of corals by depth on the far left side. And so you can see in Wilmington Canyon, we saw corals as shallow as 500 meters. That's on the one of those lines on the far right, all the way down to over 1,600 meters. And different kinds, almost every kind of coral was represented in Wilmington Canyon. So, okay, great. Now we know something about the distribution of those canyons in Wilmington. What about across all the canyons? In the next graphic here, here's all the canyons. There's 7 canyons here from Accomack, Carteret, Leonard, across the top by depth. And now look at the distribution of those color squares. And you can see immediately that there's some canyons like Accomack and Carteret and Leonard on the far left that seem to have fewer coral types there, whereas the other 4 canyons have really lots and distributed over a long range. And so you might say, well, gee, maybe you didn't look hard enough or wrong spot, or, maybe you only did 1 survey, in Accomack and you did 10 surveys in Wilmington. Well, the reality is right. It's a good question to ask for all you scientists that are watching. If you click on the next slide, I'm going to show you the depth range that we surveyed with this towed camera system. And so you can see in Accomack, we just had 1 tow, and it was shallow, 500 to 800 meters, down to half a mile. We did find corals down there. Soft corals were down there. But you look in Carteret, the next canyon over, and then we did 4 surveys there that spanned 500 down to 1,800 meters. And still, we only really saw corals at about 1,200 meters. And so it's clear to me, if you look across the breadth between 500 and 800 meters, there's very little in the way of coral that we saw. We saw a lot of sedimented habitat. This is the head of the canyons, the highest part of the canyons, loads of sediment there, ready to fall down into the canyons and get dispersed and disperse all that organic material. But as you got to 800 meters, down to 1,700 meters, lots of diversity, but still lower in Carteret and Leonard. And so even we saw corals all the way down to 2,000 meters. If you look at Lindenkohl, for example, in the middle there, you can see, or even Spencer there, Spencer, there's almost continuous coverage there of our surveys as well as the corals that we observed. So almost everywhere we looked in Lindenkohl and Spencer, we saw corals. So that's what could explain this observation of having less corals in those three canyons and more diversity and broader range of corals in those 4 canyons on the right. Well, go to the next slide. And here's one hypothesis, is that as we started out with, the 4 canyons on the far right insize the shelf. They cut into the shelf, allowing more nutrients and productivity and downwelling currents to come down into the canyon and bring nutrients, as opposed to the other 3, which were much shorter, much smaller canyons that don't incise the shelf. So our hypothesis is supported here. This is the case. Unfortunately, when you take this hypothesis up to the New England canyons, it doesn't hold up. We see a lot of diversity in the non-incised shelves. We've got more exploration to do to understand really what's happening. The other hypothesis is really it's habitat based. There's really more habitat for deep water corals in the incised canyons than the non-incised canyons. So there's more. So I'll put this graphic up here too to show you. Same sort of structure, depth from the far left, coral types by color, but across the top, instead of there being canyons, there's different types of habitats. So we looked at 9 different types of habitats that the corals were attached to. If you look on the far right column to the vertical walls, that had the most. Look at that. The distribution of vertical walls hosted corals almost every time, hard corals, especially solitary cup corals that look much like mushrooms, all the way to large colonial ones. But you can see that the vertical walls had the most coral, as coral habitat for most corals, and the tops of those walls also served as habitats. Sediments were also high when it came to things like sea pens that really like sediments, and boulders also had a lot. So it could be just the availability of the habitat and whether these things are incised or not. It's also going to be what nutrients are available and what's raining down to them, what's being brought down to the corals, and what's coming up from the bottom as well. And so next slide, please. I want to make sure that I have a few minutes anyway to discuss I just told you about the great diversity that's here, how slow growing these corals are, and how they are the foundation species for providing a linkages to the productivity in and out of a canyon. But they face many threats, and they're pretty serious threats. First and foremost is ocean warming. We're already seeing a change in deep water temperature and shallow water temperature around these canyons. Certainly seen them on the shelf, these changes. In fact, we've seen lobsters, for example, that are no longer in Rhode Island and Connecticut. They're actually migrating northward to go to cooler waters. And we're seeing that more even with deep water fish. There's some accounts of that. And so temperature, we have a changing ocean, folks, and we're not gonna stop that from happening right now. And so what would be critical for us to do is to understand how that ocean warming is gonna change where life in the canyons in our deep sea off our coast is going to be. Are the corals going to shift their distribution? Are the animals living on them, are they going to be able to shift their distribution and survive? We don't know. But we need to get out there with baselines first and foremost to understand where things are in projects like this one, supported in part by Marco, the Deep Sea Research and Technology Corals Program, and as well as the Gordon and Betty Moore Foundation. It was made this all possible because it's the first insights into this. But ocean acidification is another one. We've got we got decreasing pH, increasing CO2 going into our oceans. Oceans are absorbing over a third of the content of the CO2 that humans are putting into the atmosphere. What happens if it gets too acidic? These corals have calcium carbonate skeletons that grow so slowly, they won't be able to grow. They'll actually disintegrate or dissolve faster than they can actually grow them. So all that coral foundations are lost, all that diversity is lost, and that's a serious issue. We also have extractive industries, and that's why I had this picture, the Deepwater Horizon, where we also worked on deep water corals associated with that. We did see damage to coral life due to that spill in the Gulf of Mexico 10 years ago. And we also have extractive industries like bottom fishing, trawling of the bottom. That's a picture in the bottom right off of Australia, actually. When they brought up all that is stony corals you see in there. It could be thousands of years of growth taken away in a day. And we also see a lot of marine debris. And of the 40 canyons we've surveyed to date, we've seen trash, human trash in each one of them. Plastic, metal, things left over from fishing, like you see in the top right, like nets and traps. We see that a lot, and at varying depths. So anyway, I want to make sure we have some time for questions. I'm going to go to the next slide and then wrap up. I want to talk about sort of the future, what we're going to do now and what's important to do now. We need more exploration. If I could tell you, our surveys were only, maybe a kilometer long, less than a mile long, each one of them, most likely yielding about 2,000 images. It's hardly really a robust way of systematic exploring and learning what's there. If I were to put a dot on this map inside these canyons for the length we covered, you wouldn't be able to see it. It's tiny. And so we need more exploration to know what's there so we can look at the patterns and make our hypotheses and try to discover more and give more information to management councils and whatnot for their decisions on how to manage these resources. But we also need to examine the role that canyons play in really controlling, sustaining biodiversity. And that is what maintains these corals and the health of these corals and the health of the animals that live on them. We also need to understand, I just told you that canyons are not all alike. They have different species in different canyons, different diversity at different depths. And so what's in Wilmington Canyon or Washington Canyons, not what's in Accomack or Leonard Canyon. And so the question we have now then is, does Washington Canyon feed other canyons? Are they the source of larvae and diversity for other canyons? And do Accomack and Leonard Canyon, which are smaller and don't have the diversity, are those sinks? Do corals go there and really don't propagate out and become part of the diversity for the future? We can understand whether that's happening or not if we can get to these places and actually get physical samples of these animals and look at their genetics. And if they're not getting out, if they're not providing they're not providing diversity to other canyons, then they may actually be generating new species. These things are living in isolation. They can't escape the canyons. They're just evolving in situ. And that's why we have so much diversity in these canyons, is the canyons are helping to form or generate these new species. And lastly, I've already mentioned it, but we need to really understand the impacts of climate change. It's phenomenal that we're already seeing shallow water species move, with some people talking about they're seeing deep water fish move northward. And so if we're not out in front of this, we're not going to know what's going to happen and the changes that are going to occur and how they're going to impact this diversity, which ultimately impacts humans and our food. Lastly, you can learn more about all the things I've talked about today in a report that's available on the MARCO website. It's at midatlanticocean.org. And so please check that out if you will. And that ocean story they put together too, that goes canyon by canyon, as does a report about what we found there and what we're learning. And so lastly, I really want to thank a lot of people, and certainly the chief scientist of the missions. We have Martha Nizinski with NOAA Fisheries, Taylor Heil and Luke McCartan, who were here, who did a lot of the image analysis and work on this, merging the navigation we had from the vehicle to find out exactly where these corals and all this diversity was or is. And certainly want to thank the NOAA Deep Sea Core Research and Technology Program for their initial support to make those missions happen at sea, the captain and crew of the Bigelow, and the technical team that's with the camera system, and certainly the support given in part by MARCO and the Gordon and Betty Moore Foundation to make today possible. And thank WHOI and MARCO for hosting this event today. I really appreciate it. And now I'm ready for any questions you might have. Thanks so much for listening.
Wow, that was fantastic. Thanks. I consider myself fortunate to be able to hear you talk about this repeatedly. You know, I've heard you give presentations like this many, many times, and I learn something new every time. And by the look of the questions we're getting, a lot of people are picking up, are getting a lot of things. But I want to exercise moderator's prerogative and ask a quick question of my own first. You know, the fact that I learn something new every time, I think, is not an indication of my lack of attention to what you're saying. But you said something repeatedly when you were talking about some of the organisms that live on the corals. You kept saying, we think, or we hypothesize, or we don't know. And I was wondering if you could just speak a little bit more to that level of newness to science and what you're just learning and what there is still yet to learn that you think you can learn, or what's even beyond that.
Yeah, that's a great question, Ken. And I think, you know, we're still in the exploratory phase. This is still Lewis and Clark on U.S. land. It's just underwater. And we get such little time to be down there, to be able to see things, you know, in real time and look at the associations of the animals, like you mentioned, that we don't know. We're just identifying the patterns, and it's so exploratory. You know, last year we had 3 dives in one of the canyons in the Northeast, and we may have had 4 hours per dive in a submarine. And we made 9 samples of corals, and 3 of them we think are new to science. There were bubblegum corals. I mean, if we go down and we have just a few hours to be able to, I mean, the learning curve is astronomical because we're learning so quickly. With each species comes a different evolutionary history, a different coevolution with the animals it lives on, maybe in what habitat it lives in. You know, it's astounding. We have so many more questions than answers, but the value of asking these questions is so valuable in and of itself. We want to know what lives there, because I'm really now starting to see, just through this MARCO work, frankly, the links between the marine mammal systems that in some of these whales dive down to 2,000 meters, okay, that they are interacting with the corals. They're scooping up sediments on the seafloor. They're feeding down there. They're defecating down there. They are sloughing off a lot of material as they go. And so I feel like there's this connection with the deep sea to the shallow water that's almost, that we can see through the lens of these canyons that we can't see anywhere else. It's astounding to me that this is just, after 28 years of doing this, thinking that I was going to remote places around the world. I was going 2 miles down, 3 miles down in submarines to an alien world. It's fascinating. But here we see, for me, for the first time, the impact and linkages that are happening. And we don't have the physical measurements yet to understand the currents that are happening down there in a single canyon to really know what's happening yet over periods of time. So I don't want to be a long-winded answer, but we're on the massive exploration phase and just its cusp. And so that's why this program was so valuable to get the initial reconnaissance, even to be talking about these questions.
That sort of jumps into a question here that that Carolyn has, and Carolyn is a middle school teacher we know in Massachusetts. She says she studied the sea star. It's a Henricia genus. I can't get the species name right if I tried, and that live on sponges in subtidal ecosystems in grad school. She asked, "Have you found any sea star symbionts on deep sea corals? I saw what looked like a hipposteria in one of your photos, but it was on the seafloor." And I think that gets to that question of, are there more connections into the shallow shelf that you might not know about in the canyons?
It's true, Ken, that we'd see both of those genera in the deep sea, in the canyons, and mostly when we see sea stars that are predators on corals. They love to eat corals, and they will sit there for what we think are months at the rate that they would feed. And they eat the coral polyps, and they go down individual branches. And so that's just in the last year, 2 years, that's become of intense interest, how things are feeding on these corals and the rates of their feeding. And it's interesting you mentioned the connection again. And I didn't mention that the largest migration on Earth occurs in the water column in the deep sea as things move up and down day and night to be in sunlight, to take advantage of things, and then to hide in the deep, dark sea when predators are coming out. But I didn't also I did show a picture, I think, or a video of a red crab. And there's a large red crab populations in these canyons, and they migrate hundreds of meters, if not kilometers a day, going up and down. They're constantly moving. And they're part of that transfer of energy and productivity up and down, the connectivity into the canyons. It's also one of the fisheries that we have, red crabs are, but so they're really important. There's more and more examples of how these things are connected. I think Carolyn's picking up on that too.
Yeah, and then we had another question here from Elva asking about that vertical connection as well. As have there been any studies done about the relationship between surface productivity and the incised or the unincised canyons? Do we know what that mechanism is?
Yeah, the short answer is there's been no real comprehensive study that's looked at that in a single canyon.
All right. Well, Elva, you got a PhD topic right there.
That's a great topic.
But I think all of this really points to something, because you mentioned that the corals are some of the slowest growing organisms on the planet in the marine environment. But really, it's a very dynamic place. Things are changing constantly in and around these very, very slow growing organisms that seem to have an anchor effect to many of the ecosystems.
It's really true. You wonder how they're able to cope with the changing, or how they're able to adapt to changing environment. And I think that they have. I don't know how they do it. There are people out there that are seeking out genes that will be adapted for changing climatic conditions in corals, and that search continues. Some feel like they found things, but the answer is I don't know. I really don't know. I do know this. We did a study on fossil coral, these single hard solitary corals, like mushrooms, and we went and sampled those along the seamounts of the Atlantic at different depth ranges, and we were able to date those and look at that and how that dating corresponded to where they were at depth and with that age of the coral to climatic changes. And it showed that those solitary corals actually migrated up and down with depth over changing climatic conditions on the order of thousands of years. So where they were 15,000 years ago versus 20,000 years ago versus 75,000 years ago, they would have check those changes over that climatic time scale. And so it's clear that corals do respond over that kind of time length. But if you change the system really fast, we don't know. And that's why I think it's absolutely critical for us. It's our backyard. It's right here. We have the technology to do it, to form baselines in these canyons and set up places where we can look at these areas over time and how they change. One of the things I didn't mention was that they're slower growing, yes, but we don't see them colonize. We really don't see areas where massive colonization is taking place and these little small corals are there. We don't see it. And so they may be seriously slow to colonize or recolonize an area, one that's disturbed, or one where they've got to migrate to somewhere else because of the climatic conditions or the oceanographic conditions aren't good for them. So,
We've had several people asking, both on Zoom and on YouTube, how are the canyons named? Because some of them seem to relate to places on land, some of them are people's names.
They're mostly, from what I know, they're mostly named after people. Like the governor of the state, I think it was Governor Carteret, I think, if I remember right. We just had a most recent name was Lindenkohl Canyon here in the Northeast.
Here we go.
Yeah, there's some of the names there you see.
Yeah.
And I can't explain. I mean, maybe Wilmington and Baltimore are obvious. I don't know. Norfolk are place names. Washington's a place name, but and then you have things like Alvin and Atlantis and Nantucket there that are named after, I think, the Alvin and Atlantis the ship and the submarine.
Yeah.
But the most recent one name was after Brian Kinlan, which is a good friend and colleague who went on these initial missions in the Northeast. He was a modeler of habitats and life in those habitats for NOAA, and he passed away a few years ago, and we honored him by naming a canyon after him. So Kinlan Canyon's up in the Northeast.
So we spoke about the canyons, and you mentioned how many of them were originally formed during the deglaciation. But Mort here on Zoom is asking, or no, I'm sorry, not Mort. John is asking, what about the seamounts? How are they formed?
Seamounts were formed from a hot spot of volcanic activity in the middle of the Atlantic. And as it's the western part of the Atlantic seafloor moved northwest over hundreds of millions of years, that it tracks that hot spot. So it created a seamount. The seamount migrated on top of the plate, the oceanographic plate that's the seafloor, and it migrated toward New York, frankly. And so these are sort of extinct volcanoes, hotspots. In fact, if you really look at the geology, that New England seamount range traverses all the way up into northern New York as mountains.
Thomas was asking also, what's the water temperature for these corals? I mean, we're familiar, I think a lot of people are familiar with maybe scuba diving on reefs and in warm tropical waters. Well, what's it like for these corals?
We see these corals at deep water temperatures of 4 degrees Celsius, 5 degrees Celsius, 7 degrees Celsius. I'm sorry, I'm not doing it in Fahrenheit. I can't think that way. It's just not my language. I'm sorry.
37, 40, somewhere.
Yeah, and so I know that hard corals like Lophelia, the reef-building coral Lophelia, really doesn't like warm temperatures. If it gets more than 14 degrees Celsius, again, I don't know what that is exactly, it really starts to die. And that's one of our concerns, is people are trying to look at the different temperatures that these corals are living at and seeing what their boundary conditions are. And so we don't have it for a lot of corals. It's very hard to rear them and keep them in aquaria in your lab. You can do it. Whether you can simulate really deep sea environment is another question, but you can keep them alive and expose them to temperatures and see how they do. But we've seen warming in the Gulf of Mexico, for example, that's impacted Lophelia reefs when they get warmer.
We've been talking about the U.S. East Coast, and I know you showed an image of a deepwater coral in Hawaii, but where else are deepwater corals found?
Deepwater corals are found all over the world. There's probably over 200,000 seamounts around in the deep ocean, and corals love to be on structures like that, like seamounts. They're on ocean ridges, which encircle the deep sea floor around the world. They're in canyons around the world. There's canyons in the Arctic. There's canyons off of New Zealand. I mean, every continental margin seems to have canyons of some sort. And so they are, we believe, around the world. And we see them as deep as 6,500 meters or so around there. So they go really deep too.
Allison is asking, the difference that you see in species, you've noted the differences in species that you saw in each of the canyons. Have you also noticed any physical or chemical differences in the movement of water or in the composition of seawater that might speak to some of those differences?
That's a great question. And people are looking at that now. We haven't done that in the canyons yet. We haven't really had that access to the data and whatnot. That's our next step, I think. But there are people who are doing it in the Pacific that have done it really well on seamounts in the Pacific, looking at different, not just temperatures, but water masses that have different salinities, densities and temperatures and whatnot, and how they're seeing a correlation between coral types and water mass types with those properties. It's often hard to distinguish between the impact of depth on a given species of coral, for example, versus temperature, because they co-vary together. As you get deeper, you get colder. Yeah. And so determining what individual factor might be controlling the distribution or range of a single species can be really difficult. But you can come up with a set of observations that can limit the alternatives, the options. So there is evidence that groups of coral species are found in certain water masses in the Pacific on different seamounts.
Are there similar canyons on the West Coast, John is asking?
There are canyons on the West Coast off of California and whatnot that have been studied quite a bit. Because of the tectonics of North America, the deep sea occurs very close to the margin there, and so it drops like it's all a canyon, and then within that drop you have gullies that become a canyon, and so they have been well studied. They have corals, they have sponges, they have lots of fish there. The deep sea is amazing. If you give the deep sea some hard bottom or even some sediments, there's going to be lots of life there. It really is truly amazing.
Life will always find a way. Yeah,
it's true.
I gave this sort of very broad litany of the places you've been at the start of the program, and I've been fortunate to be on a couple of expeditions too. One of them, a very exotic place just north of New Zealand to the Kermadec Trench, one of the deepest places on Earth, and yet, as you said, right here in our backyard, we have this amazing diversity and this amazing abundance of life. How does this place that we might overlook because it seems, it's just our backyard, how does this place compare to some of the other places you've been to around the world over the years?
Wow. I mean, that's like a half-hour answer, but I think,
I know.
Because I've studied hydrothermal vents. I mean, I've witnessed, the only seafloor eruption that was really witnessed by a person's eye. And there's been amazing places that I've gone to. I've been very fortunate. But I think to the point of your question is that these canyons, our deep sea, and that's right here off our coast, is a window to access we don't have other places, right? We have the technology, we have the ability to get to these places in less than a day, and without the great expense. And so it's a living laboratory for us right here off our coast. And the fact that we have multiple and different kinds of canyons is such a natural laboratory setup, ready to go. So I'm talking to marine mammal people for the first time in my life, and how that might interact with deep water corals and how they might supply each other with energy, productivity, carbon. I think that it's hard to compare hydrothermal vents and exploding volcanoes on the seafloor to whale carcasses on the seafloor that have a whole unique set of life on them to a canyon. I think the value here in terms of what we can learn now in the next generation of deep ocean science is dramatic. It's really, I mean, people are going to catch on to these canyons are right here, and we can do a lot of time series studies here that are going to revolutionize the way we think about how life can live in the deep ocean and on Earth.
I think that speaks to what makes them so special, is the potential for learning. One of my favorite times in a lab on a ship is when something comes into the lab straight off of the seafloor, and people like you look at it and go, "What's that?" We've
done that many times. I remember there were 28 biologists on board. We brought something up from 7 miles down, 10,000 meters in the Kermadec Trench, where you were there, Ken. And these are the world's leading biologists. It felt like, and we had no idea what phylum to put this animal in, and it was ridiculous. I mean, I think that's why I do it, from the sense of discovery and amazement. So,
and you can do that right here. As you were saying, you discovered three new species just on a handful of subdives just 2 years ago.
Yeah, and we were myopically just looking for corals too, and the different corals that were there. We weren't looking for all the other things that are there. And we know that species in the sediment that we can't see are really diverse in these canyons. Didn't even really get to that, but all this organic material that's landing on the seafloor and going deep into the sediments, they're thriving down there because of that productivity. And we really need to get a better handle on how those things are all related.
We've got time for a couple of questions. Just quickly, Elise is asking, what can people do help these canyons, to help them thrive, and just that? What can people do?
You know, I would say, you know, the human impact in the canyons can be seen visually by just the amount of trash we've seen, the plastic that we've seen, the plastic bags and magazines, and I've seen toilets and showers, and I've seen airplane engines, and there's all kinds of cans of all sorts, you know, soda cans. I mean, it's just phenomenal. Yeah. There you see there's a piece of black plastic there in the center of the screen on the sedimented seafloor. And then we see lots of nets. It's just how it is. And, you know, I don't, what do it, you know, don't litter, you know, don't, you know, I think, you know, we could talk about not littering or stop the stream of plastics or whatnot. And that's always a mindful thing to do. But I think we need to change the way we think. If you can do that, if you can influence people that make decisions about funding research, about learning more about these things, about people that want to make television programs about this, to educate people about our ocean and how it impacts us every day. I'm learning more and more about that all the time. I used to actually think that the deep sea was remote and it didn't impact us at all. And now I know that the deep sea is coming up to meet us in the shallow waters off our coast here in these canyons. And I know that the food that I'm eating at dinner came from near or in these canyons, Jonah crab, red crab, whatever, that's, you know, that's vital for us. I get that. You know, I'm not a scientist who's advocating any which way except for information. My goal here, my job here is to try to give information to people to make good decisions, to educate you, the public, about what we're learning. I'll learn with you as we go, and be more intelligent about the decisions we make. If you value the deep sea, your oceanography, your oceans at all, that control your daily weather and your climate every day, then behave like it. Make it part of your life. Make yourself aware of it.
Last question: what's next for this data and for the work that you're doing?
And so these data are tremendous, because what we can do with this depth, location of corals by depth, and by the maps that we showed you, we know the slopes of the habitats the corals are on. So we know now from this work that if a slope in a canyon is over 45 degrees, we're really likely to see corals of a certain kind. We see vertical walls, we're likely to see hard corals and certain soft corals. And so we can put those in models now and predict where we can't go and explore, but we see these slopes, these vertical walls, that this kind of corals ought to be there. And by predicting that, we can say, hey, what should it look like? How many corals are really down there? How much are they producing? How much are they giving to habitat to the other animals? I mean, it's a launch pad. These data are a launch pad for predictive modeling of where things should be for biodiversity through the corals, and what our plans should be next to explore, to where do we go to. I mean, I want to go there and sample corals and look at how they've co-evolved with the animals living on them. I need physical samples of the DNA to do that. And so where am I going to strategically target where to go? And changes with depth I'm going to look at, and across these canyons. And so, you know, if we can model where they should be, that's a much greater chance of getting to these corals and making really massive discoveries.
All right. Thank you, Tim. I think that's all the time we have for questions today. Before we close, I want to say again a big thank you to Kisha and to Marco and to Tim for sharing your thoughts and your insights with us. Especially thank you to Marco for helping make this happen today, and to the Gordon and Betty Moore Foundation for their continuing support of Tim's work and the important work that Marco is doing. And I want to thank all my WHOI colleagues who helped make this happen, who've been working behind the scenes. Finally, Craig, if we could pull up that last slide. If you want to learn more about the canyons off the east coast of the US and their importance to a healthy ocean, you can visit Tim's lab site, shanklab.whoi.edu, or the Marco website, midatlantic.org. You can also visit the WHOI homepage, WHOI.edu, for information about the ocean in general. You'll find more resources both at the WHOI website and at the Marco website like I said, general, but also specific to this topic about the Mid-Atlantic canyons. That's all the time we have. Thank you everyone for joining us. And once again, stay safe, stay healthy. Thank you.